Hi everyone, my name is Shiran. I'm finishing my PhD studies in Tel Aviv University under the supervision of Professor Itai Miroz. I'm a bioinformatician, and in this talk I'll tell you how we can use machine learning to modify our genomes. I'm going to talk about the CRISPR system. So what is CRISPR? CRISPR was found in bacteria several years ago and was adapted as a genome editing technique for more complex cells like human cells. Ever since CRISPR was found, we hear titles like "Scientists have eliminated HIV in mice using CRISPR." CRISPR is going to revolutionize our food system. Cancer cure close, gene editing therapy could cure cancer and all inherited diseases in the next 20 years. And the genetic breakthrough that could change humanity. What's all the fuss about? So CRISPR targets the DNA. In each cell of our bodies, there is a long sequence called DNA that is similar to beads on string. The DNA is made out of 4 types of beads, 4 types of molecules, which we denote as A, C, T, and G. Now, this long string of the DNA is built of some kind of an order of those A, C, T, and G characters, molecules. Inside those long DNA strings, we can find substrings, subregions that are called genes. The order of the molecules A, C, T, and G in a gene dictates the function of this gene. For example, one gene may code for cholesterol, other for digestion hormones, etc. Overall, most of our genes, humans, are very similar. There are small differences that make us different from one another. And in some cases, a mutation in a gene may occur, and it will cause it to malfunction. For example, it may cause it to generate a cancer tumor. Now, how can CRISPR help? So the aim of the researcher is to direct CRISPR to the target gene in the DNA. In order to do that, the researcher designs a short sequence that is very similar to the DNA. It's called sgRNA. And he has to design it such that the sequence is very similar to that of the DNA. When the researcher inserts this CRISPR with the designed sgRNA into the cell, the CRISPR sort of scans the genome until it finds a match to the DNA. Once there is a match between the sgRNA and the DNA, the CRISPR cleaves this gene and causes it not to function anymore. This is great, because if we know the mutation that causes a disease, we can design the CRISPR to cleave this gene and stop it from functioning. So if you think about it, we have a cure for all mutation-caused diseases. Great, but there is a problem. It doesn't have to be a perfect match. CRISPR is very specific, but it has errors. We might design an sgRNA to target one gene that causes a disease, but it will also target and cleave another gene when there is a mismatch between the CRISPR design and the DNA. And this other gene may be very valuable, like for eyesight, for digestion, for hearing, whatever. So due to this problem, much research has been done trying to identify which sites in the genome are cleaved that were not supposed to be cleaved when applying CRISPR. Some of these studies were done in a genome-wide manner. That means they applied a CRISPR design to the genome and then scanned the whole genome and reported the sites that were cleaved and the frequencies at which they were cleaved. So, of course, they reported the on targets, the design targets that are match exactly the sgRNA that they designed, here in green, and the frequency at which they were cleaved. And also the targets, the off targets that are spread over the genome, the false positive of CRISPR, which contain mismatches to the CRISPR design. So these data help us design the tool for an optimal design, for an sgRNA, that has high frequency at the on-target site and low efficiency at any other sites. That means minimum off-targets. So in this study, I collected several studies that did this genome-wide off-target detection, together with the sequences and the reported frequencies. Since these studies were made in a genome-wide manner, every possible similar sequence that was not reported could be manually found in the reference genome. And so I detected those and added them as the uncleaved sites as the negative samples. From each pair of sgRNA and the reported or the uncleaved site DNA site, I extracted three kinds of features, ones that are specific to the DNA, ones that are specific to the sgRNA, and ones that describe the match between the sgRNA and the DNA, like mismatches. From all of these, I trained a random forest regression algorithm to predict the probability of cleavage according to those features. Altogether, there were 32 sgRNAs with a reported 715 cleaved sites, tens of thousands of uncleaved sites, and 80 features that illustrate those. The trained machine learning algorithm is called CRISPR Target Assessment. Now, how accurate is CRISPR? So I examined this in cross-validation and of course on a validation set, but in cross-validation, we see that the accuracy, the Pearson R-square between the observed and the predicted cleavage frequencies was 80%. Compared to other tools, very popular tools, we see that CRISPR is better than the others. CRISPR reaches an 80% accuracy, while the next best is 65%. So the question is, what makes CRISPR more accurate? There are several factors, but I'll focus on the most interesting feature that was taken into account in this study. Previous tools focused on the match between the sgRNA and the DNA, the mismatches between them. But if you think about it, I told you before that the DNA is very long. It's about 2 meters, and it's folded in a compact manner into a single tiny cell. So the DNA has a 3D structure. Now imagine that you're holding a pasta stick. Now try to break the pasta exactly in the middle. It's very easy. Take this pasta and cook it. Now, again, try to break it exactly in the middle. It's more of a problem. You have now to spread it and then tear it. Now take it to the other extreme. Imagine that it's a metal pole. Now try to break it, you can't. So with CRISPR, it's the same deal. When it's too rigid, CRISPR can't break it. And when the DNA is too flexible, it folds over other elements in the cell, and CRISPR can't reach it. So the features in my study show that the rigidity in between this span between the most flexible to most rigid is the optimal for CRISPR cleavage. And with that, I would like to thank my supervisor, Itai Meiroz, and my lab mate, of course, to the colleagues that worked with me on this project, Professor Frank Zhang, Winston Yan, and Didi Amal. Thank you to the funding sources, and thank you all for listening. Please feel free to contact me for any question.